Before we start today, any questions? Now you've had your first Wednesday review session. You good? Any questions about it? About other stuff? Yeah. Okay, question was, do I post past or practice exams? And the answer is no. And the reason is because that's the idea of these multiple choice questions that come up in the review session. Any other questions? The tricky thing is, so about past exams, the idea is that Brittany writes half the questions, I write half the questions. If I gave you an old exam, it would be a different person. It would be similar, but different. So that's why we try to just do the ones for the Wednesday. Any other questions people have? Okay. So what I want to start with is this is what we're going to get into today. But before we get here, I want to wrap up inbreeding that we were talking about last time. And in particular, this is sort of the one point that I didn't have quite time to get to last time. And that is, I want to look at the relationship between inbreeding and population size. So, and the reason for this is in small populations, individuals, in small populations, if you choose a mate at random, it's quite likely that that individual is related to you. So random mating in small populations can lead to inbreeding. That's why I want to discuss it. And I'm going to give you slightly a hokey example, but I think one that makes the point. And that is inbreeding depression is a big concern in zoo animals. Say you're a zoo, and a zoo has two camels, a male camel and a female camel. Over time, camels doing what camels do, that zoo may have 6 camels, right? So now the zoo has a population of 6 camels. If you've looked at where camels live in zoos, they tend to live in these sort of big grassy enclosures with a few rocks, right? What's to stop any given camel from mating with any given other camel in that zoo? If that happens, you're going to have inbreeding, right? Because 4 of these camels are full sibs. So basically, this is an example of obviously a very small population of how inbreeding can happen in small populations, even if an individual chooses to mate at random. You can think of a population of, say, 20 individuals. Even if those individuals are not related to each other now, this generation, a few generations down the road, if the population stays about the same size, a number of them are going to be related to one another. So in small populations, random mating can lead to inbreeding. And this is, as I was implying, a big issue in zoos. I'm not going to go through this table, but just suffice it to say that what zoos do is, internationally, zoos keep these stud books, and they control which animals mate with other individuals, and often include matings between individuals in other zoos to try to reduce inbreeding depression, which historically was a big issue in zoos. You can imagine in conservation, this is a big issue. And what I want to do is wrap up this section by going through an example. And the reason I want to spend the time to go through this example is because in small populations, I've said inbreeding is an issue. And then lecture before last, small populations, drift is an issue. You got a small population, and it's not a small population, and it's not either or. Both of these things happen at the same time. And what I want to do is just work through an example for you to allow you to see how this happens. So this is a Florida panther. You're sort of member of the North America. North America has a number of panthers, or they're also called pumas. This represents the historic range here. This is the current range. What you can see is right now it's living in a minuscule portion of its original range. And this is due to mostly loss of habitat. Some was hunting. And the population got down to about 20 individuals. Given that around half of those 20 are male and around half of those 20 are female, I mean, that is a really small population. And not surprisingly, well, so that's a really small population size. Once people realized that things were that bad, they stopped hunting, they started protections. There's a lot of actually issues in Florida with panthers getting hit by cars at night, so they just changed how traffic was moving, blah, blah, blah. Protected these panthers, nothing happened. Population size stays the same. What's up? Well, what's up is that these Florida panthers were suffering from a number of genetic anomalies. What this is showing here, this is all in your textbook over here, is defective sperm. So these are rates of defective sperm in the Florida panthers, around 40%. These are rates of defective sperm in other panthers, so relative. So you can see it's doing worse than other panthers, and then compared to other big cats. So clearly there's a problem here, and it wasn't just the sperm. These panthers had heart defects, undescended testicles, skeletal malformations, little end of the tail had this little, bend in it. Anyway, just a lot of genetic anomalies. What does it tell you? Given what we've been talking about in class, it suggests the expression of deleterious recessive alleles, right? And in fact, if we look at genetic diversity, so if we look at genetic diversity, here's genetic diversity in big cats, other North American panthers, and then the Florida panthers. So reduced genetic diversity, what does this tell us? It tells us a bottleneck, genetic drift, something like that, right? So we have some evidence of inbreeding with the expression of these recessive deleterious alleles and reduced fitness. We have some evidence of drift by this reduced genetic diversity. And now I just want to sort of stop back and say conceptually what happens in a small population. In a small population where you get a founder effect, you get genetic drift, what can happen is that recessive deleterious alleles can actually wind up increasing in frequency due to genetic drift, right? Because of these sort of founder effects and really small population, really strong drift. So recessive deleterious alleles can increase in frequency. So when inbreeding happens, all of a sudden the magnitude of inbreeding depression can be a lot larger because these deleterious alleles are no longer at low frequency, but some of them are at higher frequencies. So this is an example of sort of the combined effects of inbreeding and drift that can lead to real problems in small populations, right? Because the population is small, so you get drift, you get the increase in these recessive deleterious alleles. Inbreeding happens even due to mating at random. You get inbreeding depression, you have individuals die. So did that small population grow? No, actually, maybe it's shrinking. So that when populations get really small, they can get stuck in this sort of vortex where the size winds up getting sucked down due to this combination of inbreeding and drift. So when they protected the panthers and the numbers didn't go up, that's what people thought was going on. And so what they decided to do is sort of a conservation effort to mitigate this problem is that they brought in some panthers from Texas. Small number of panthers from Texas into Florida. Like a small number, like 2 or 3, I've forgotten exactly. And then so this figure here shows pure Florida panthers. They were here, we can just ignore that. What I want to show you here is this is the survival. Survival of Florida panthers, not so good. They bring in the Texas, they mate between Florida and Texas, and look at this, the hybrids do great, okay? These recessive deleterious alleles that are high frequency in that Florida population are again hidden when they make heterozygotes with the Texas population. And so they bring in these Texas ones, heterozygosity goes up, population size starts to increase, etc. So how effective, the fact that it was so effective to bring in these Texas panthers suggests that they actually diagnosed the problem right, that the small population size was leading to this drift in inbreeding, and by bringing in some genetic diversity from elsewhere, that was increasing the effective population size a lot and letting these panthers sort of escape this downward spiral. Anyways, in your textbook, a really nice example. You can imagine how this is not isolated to the Florida panthers, but it's something that we have to think about in conservation all the time. Okay, any questions about that example before we go on? All right, so now moving to topic of the day, and that is, so far we've talked about sort of these Hardy-Weinberg assumptions: selection, drift, random mating, and today we're going to pick up the last two, mutation and migration, but instead of talking about them in isolation, what I want to do is pick up the theme of this last example, and that is often different evolutionary forces act in concert, right? So in a small population, you have drift and inbreeding occurring. It is very common to have different evolutionary forces working together. And so when I talk about mutation and migration, that's the context that I'm going to talk about it in. And in particular, the context is looking at how much genetic variation is out there. Okay, so that's the sort of how we're going to bring together these different evolutionary forces. Okay, so genetic variation. Let me see. All right, if you think about it, if you have selection happening on a trait, let's say that selection of a mouse living on a beach favors tan fur. Over a lot of generations, you expect all mice to be tan and all alleles to be tan, right? Selection that is in a direction you expect is going to wind up increasing the frequency of the favored allele. The less favored allele decreases in frequency. And so over time, we expect that selection is going to act to reduce genetic variation, right? That dark allele, if you live on the sand, is bad. It's going to disappear. So selection, as I sort of intimated before, is this pervasive evolutionary force. It gives rise to all the biodiversity around us. It's everywhere, etc. But if selection is active to reduce genetic variation, then we're left with this question of evolutionary biologists, then why on earth is there so much genetic variation in the world around us? We can see a lot of genetic variation at the level of the phenotype, right? In this room we have practically all of our sort of quantitative attributes we have variation for. We have variation at the level of the nucleotide sequence. There's just genetic variation everywhere. So if selection gets rid of genetic variation, and there's a lot of genetic variation out there, the question is why? Turns out this is actually a little bit of a complicated question, and there are two basic answers. One is there's a lot of variation out there, and it's irrelevant. In fact, I have brown hair, somebody have blonde hair, somebody have black hair. It doesn't matter. It's neutral as far as selection is concerned. We're going to talk about that later after the first exam. So that's when we get into molecular evolution. The other possibility is that there is so much genetic variation out there. The other possibility is a set of selective hypotheses, meaning that selection actually doesn't just get rid of variation, but it also helps maintain variation. And so what we're going to do today, and maybe spilling into Monday, is go through this laundry list of 5 different mechanisms of how genetic variation can help how selection can help maintain genetic variation. Okay, so we're going to start off. The first one is looking at mutation. Remember, only mutation creates new genetic variation. One possibility is there's a lot of genetic variation out there because there's a lot of mutation out there. But the data doesn't really bear this out. Here is a very old table of a bunch of mutation rates. I think I kind of like it because there's a bunch of different organisms here, going from bacteria to people and plants and animals in between, and these are phenotypic traits. But the big thing to notice is that these mutation rates are really small. 10 to the minus 4, 10 to the minus 6, 10 to the minus 8. These are mutations per locus per generation, right? So these are really small changes. Here's a figure from your textbook. This shows if you have a mutation rate of 1, 2, 3, so this would be 10 to the minus 4, from big A to little a over 1,000 generations, always big A to little a, you get change, but not what you would call a massive amount of change, right? This is 1,000 generations. So, clicker question number 1. What do you think this tells you about this? Yeah, okay, this is what we did from the questions we had last time, right? Looks like about everybody is in. And it looks like almost everybody has chosen C. And C it is. But let's talk about C for a second. So this figure suggests that mutation is a weak evolutionary force. Yeah. Exactly. So mutation is really important because it's the source of new stuff, novel genetic variation, right? But in terms of actually creating much evolution at the level of the phenotype in a population, it's a weak evolutionary force. 1,000 generations to get from 0.9 to something like 0.8, that's pathetic, right? Selection is incredibly more powerful than that. So yeah, that's the deal with mutation. and it creates genetic diversity. And one possible, and it creates a mechanism for why we see genetic diversity in populations. And I want to develop this idea by talking about something called mutation selection balance. And I want to deal with this first just at the conceptual level. I'm going to write on the chalkboard, but I think it's all here. So at your A locus, so say your big A allele, U is the mutation rate. So this is a mutation from big A to little a. And it happens with the first species. Little a is recessive and deleterious, as we've discussed, most mutations are. When it is expressed, and remember that's going to be in the homozygote, right? Little a, it is going to get removed by selection. All right, so big A leads to little a through mutation. That ups the number of little a's in the population. If you get enough little a's, it's going to get expressed. Just two heterozygote individuals are going to mate. You're going to have little a kid. If little a is bad, those little a's are going to get removed by selection. So there is some frequency of little a in that population where the number that are being added by selection in any generation are equal to the number that are being removed by selection. Okay? So we call that the equilibrium frequency, right? Because equilibrium means not changing. So the amount of little a isn't changing. New ones are being added by mutation, and they're also being removed by selection, and you get some sort of standing frequency of little a. So what does that do? That gives you a predictable amount of genetic variation in your population, this equilibrium frequency. And what I want to do in the next couple slides is talk about sort of what this predictable frequency is. Naturally, we go back to good old Hardy-Weinberg, except this time you're not expected to work through all this stuff. I'm going to show it to you quickly so that you know it's not magic that I came up with this, but all you need to know here is what happens at the end. Okay, so we got two evolutionary forces we're talking about, mutation and selection. We've talked about selection in Hardy-Weinberg before. This is a little refresher. The frequency of big A after a round of selection, or P prime, is the frequency of big A after selection, plus half of the frequency of the heterozygote after selection. No problem, right? Then we just say, okay, we know what this is, and that's what I plugged in here. The frequency of big A after selection is P squared times its fitness, divided by W bar. Heterozygote 2PQ times its fitness, divided by W bar. We've been there before. Now we get to the tricky part, because we know, the way we set this up, we know something about fitness, right? Because we have said we're interested in a recessive deleterious mutation. If it's recessive and it's deleterious, we know the fitnesses, right? We know that the fitness of the heterozygote must be the fitness of big A, because little a is recessive. And we know that little a is deleterious. So we know that these guys are 1. So you can just plug 1 into this equation, and it gets carrier looking. Why does it get worse? Because the W bar has been expanded here. But the bottom line is, and that's what's down here. But you can skip this intermediate step. I'm just showing you can get from point A to point B if you want to. And let's assign S to be 1 minus W big A. So remember, W little a is 1 minus S, or S is 1 minus, or the relative fitness of little a is 1 minus how bad you are. Anyway, by plugging these two in, the whole thing, by plugging this in, and knowing PQ equals 1, you can miraculously reduce all that chaos down to this lovely small little thing. And now we do one more trick. What I'm about to show you is we're getting to the world's easiest equation through all of this hairy math. At equilibrium, there's no change in the real frequency, right? So we're talking about equilibrium conditions. So at equilibrium, P prime and P are the same, right? Because there's no change in the little frequency. The amount of P next generation, the amount of P this generation are the same. So you can set these to be equal, and, oh, this just indicates that we're talking about equilibrium, blah, 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 and voila, you get this lovely, lovely, simple little equation. Now that we've kind of taken this path to get here, what does this equation tell us? It tells us that at equilibrium, when both mutation and selection are acting, the frequency of the recessive deleterious allele is simply the square root of the mutation rate over the selection coefficient. Let's think conceptually about this to see if this makes sense. The selection coefficient is how bad you are, right? A big selection coefficient means the allele has very strong negative effects on fitness, i.e. it's bad. So if this gets bigger, this gets smaller, right? Your denominator is bigger, your fraction is smaller, your frequency of Q is going to be smaller. Intuitively, that should be comfortable, right? So the more selection is acting against this allele, the lower its frequency are going to be. Same with mutation. Here we have the mutation rate here. If the mutation rate goes up, so more of these little a's are created, then it's in the numerator, the frequency of Q goes up. So on a test, if you get this backwards, just think through it. And you're like, you start freaking out. Did I get it upside down? Did I get it right side up? It's easy. Just go through it intuitively and see if it makes sense to you. Okay. So, one thing, your textbook goes through a couple of examples that use this, and I expect that you should be able to use this. But one thing I'll say is that historically, this equation has been really useful to people. So before we were able to get kind of a molecular access to what mutation rates are and understanding molecular genetic variation, this was done using the phenotype. So if we're trying to understand human genetic disease, for example, with human genetic disease, there was a good sense of selection. So what were the survival and reproduction rates like of people that had different human genetic diseases? And they had a sense of what Q was because they knew how often they knew the frequency that people expressed human genetic diseases in populations, and that would be little a. And so this was some of our earliest information that we had on what mutation rates really look like. And so over sort of the history of biology, this has been actually a really powerful equation in telling us things that now we have a molecular access. We have a lot more data that confirms what we were able to get just via the phenotype. Okay, that's mutation. Now I want to turn to migration. And migration is another way, you can think of it like mutation. It's not random, I put these guys together. Because migration is another way that genetic variation enters populations. In this case, instead of it being created brand new, the genetic variation enters a population from somewhere else, another population or a number of species. So when I say migration, what I mean is genetic movement, not like birds migrating south in the winter and migrating back north in the summer. That doesn't necessarily affect the genetics of the population. It's the entry of new genetic information. Okay, And for migration, I want to introduce, this is a classic population genetic model called the continent island model. This is very commonly used in conservation biology. Why is it commonly used? Because it's called a continent island model, but you can sort of extrapolate to a more general context. A big population, a small population. If this population is small, say we have 10 individuals moving from the continent to the island. If the island has 50 individuals on it, 10 individuals moving is going to make a big influence on the allele frequency, right? If you have 10 individuals moving from the island to the continent, but the continent has like 3,000 individuals, those 10 are barely going to budge the allele frequencies. So even though you can have individuals moving in both directions, because of the size of the population is so different, this is the migration that winds up really counting. So we're paying attention to this big arrow here. And so you can see why it's used commonly in conservation biology. You may have a large, stable population, and then some satellite populations that are smaller that people are worried that are going to disappear or weak out. Okay, so in this continent island model, we're going to stick with our sort of our A locus situation, but what I want to do is allow the continent and the island to have different allele frequencies, right? So we have some allele A, and it's got a frequency of PC on the continent, and it's got a frequency of PI on the island, and the M is the migration rate. So it is the fraction of the island's population that came from the continent. Okay, let me write that on the board. So M is the fraction of the island pop from the continent. Okay, so that's the setup, and now there's all kinds of things that we can do with sort of continent island migration, but we're just going to do something very simple and first say, with this information, what is PI? PI would be the frequency of big A on the island, and here's the answer. Looking at she's like, oh my god, what is this? But this is simply PI, that is how common big A is on the island, depends on two things. How common big A was on the island last generation, T minus 1, and the fraction of the population that was island individuals, and how common it is on the mainland, and the fraction of the population that came from the mainland. So if you think about these two pieces, they're pretty straightforward. This is PI includes some individuals from the mainland, how common are they, and what's their allele frequency, some individuals from the island, how common are they, and what's their allele frequency. That tells you what PI is on the island. So this shows you how migration can change allele frequency. And migration can be a really, actually fairly powerful evolutionary force. Migration, mutation is changes one like nucleotide or one SNP. I mean, mutation rates can be very localized. Here you're bringing in a whole new genome, a whole individual. And there's a rule of thumb that if you have about one migrant per individual between populations, it can reduce evolutionary divergence. Okay, yep, so that's going on there. And now, this is where I'm really getting to the maintenance of genetic variation. Just like we talked about mutation selection balance, we can have vibration selection balance. And that is some equilibrium number where the number of new big A's being brought in by migration are the same as the number that are being removed by selection. we're not gonna work this out mathematically, but instead I want to give you an example because I think this example is great at getting the idea across. Water snakes in Lake Erie. You may think random, but it's just a really cool example. Here we have Lake Erie, which borders on continental US and on Canada. Here we have Ohio and Ontario. These are the continents, right? We got a bunch of islands in the middle of Lake Erie shown here. And these water snakes occur in these sort of beautiful variation in banding patterns. Some water snakes are just plain solid. Some of them are highly banded, and some are a little bit sort of more diffusely banded. The banding is a one locus, two allele genetic basis. All right, so if we look at what's going on the continents, we find that of these four genotypes, four phenotypes, sorry, non-banded is A, highly banded is B, and sort of B and C are intermediate. What we find is on the continent, almost all individuals are this really strongly banded pattern. Doesn't matter whether we're talking Ohio or Canada. But if you look at the islands, you see something really different, right? The islands have few banded and everything else. And in particular, if you look at these islands, Middle and Pele Islands, they're the ones furthest from the shore. You have the highest frequency of the non-banded individuals. So in different spatial areas, we have different phenotypes reflecting this sort of one locus, two allele. It's exactly like that continent island model I just showed you, right? The frequency of the banded allele is high on the continent. The frequency of the non-banded allele is higher on the island. What's going on here? Well, it turns out that these islands in Lake Erie are surrounded by these sort of large limestone rocks where the snakes, in particular the juvenile snakes, like to go and bask in the sun. And if you are an unbanded snake, you blend into the rock and are less visible to predators. So there is selection against the banded alleles on the island because of this rocks. The mainland, I guess the mainland doesn't have these rocks, it just has a different selective environment. So then you might say, well, if it's good to be unbanded on the islands and good to be banded on the mainland, why do we see all of this sort of chaos going on these islands with these different banded phenotypes? And the answer is snakes swim. And they, in fact, swim from the mainland out to these islands. People in boats see water snakes. They're called water snakes, okay? So they see these water snakes swimming through Lake Erie, and some fraction of them make it to the island. In fact, given this genetics here, what we know about allele frequencies, they can calculate the rate that snakes migrate. So every generation, approximately 12.8 snakes migrate from Ohio and Ontario to these different islands. So this is an example of a migration selection balance where you have the combination of migration bringing in new alleles, selection getting rid of those alleles, maintaining genetic variation on these islands for banding pattern. Okay, any questions about this? All right, well, I see we are really not going very far with today's lecture today. Okay, so that was the second mechanism for maintaining genetic variation. Next one we've already talked about, so I'm putting in a slide you've seen before just as a reminder. This is the warfarin rat poisoning, and this is resistance to the rat poison. And when there is no rat poisoning, you get low levels of resistance. You add rat poisoning, you get high levels of resistance. This is, now I'm going to sort of generalize this. We can think of this as a fluctuating selection environment. And that is the type of selection that's present changes over time. You have rat poison, it favors one thing. You remove the rat poison, the wild type has the advantage. So when selection fluctuates, different alleles are favored by selection at different times. This is a really, really common phenomenon. I mean, obviously rat poisoning is something people created, right? But out in nature, you have some years, you have dry years, you have wet years, you have cold winters, you have warmer winters. You have some plants that live in shadier habitats, some that live in sunnier habitats. So there is a tremendous amount of both temporal and spatial variation in selective environments, and as long as you have no allele that's best across all environmental conditions, you wind up keeping genetic variation in populations. Okay, so conceptually, really simple, but really predominant. All right, so now I want to talk, this is the fourth way in which genetic variation is maintained, and it is determined by the relative differences. And so I'm just going to let you guys start with this quicker question, and then we'll take it from there. Got most people in? All right. A little bit more heterogeneity than average. So most people chose C. Let's work this through. Let's first look at these gene fitnesses. Obviously, heterozygote has the best fitness, right? Then what's got the next best fitness? So S2 is bigger than S1. So you're kind of lame about it. S2 is badder, right? So this is a larger number, so 1 minus this larger number. This is going to be smaller than this, right? So the relative fitness of, so W of big A is bigger than W of little a, and then this is the highest in terms of fitness. Okay? So, if that's true, how do you expect evolution to change allele frequencies? And this is sort of in the long term. Well, if this is the worst, this is definite, little a is definitely going to be less common than big A, right? We agree with that. So the question is whether it's going to be B or C, because both of these have big A being higher in frequency than little a. And so the question is whether big A is going to go to near fixation or it's just going to be more common. And the answer is, as most people figured out, it's just going to be more common. Why is it going to go to near fixation? Because because big A and little a are both in the heterozygote. The heterozygote has the highest fitness. Okay, so the relative frequency of big A and little a is going to depend on exactly what S1 and S2 are. But the bottom line is having the heterozygote have the highest relative fitness maintains variation. It keeps both alleles in the population. This is, you're probably familiar with this as it is termed, heterozygous advantage. And classic examples are sickle cell anemia. This one is cystic fibrosis, which I'm going to go through this fairly quickly. So cystic fibrosis is a genetic disease that's caused by a loss of function mutation. Until recently, few individuals with cystic fibrosis made it to reproductive age, and yet it is really common. So 1 out of about every 2,500 European newborns of European ancestry, I should say, express cystic fibrosis, which gives you an allele frequency of like 0.02. So that's 2 out of every 100. I'm not kind of glazing over the details, cystic fibrosis. Yeah, I'll just leave it like that. It's from your textbook, which has more information on it. So really pretty common mutation, considering that until recently, it had substantial impacts on survival and reproduction. And it turns out that, or what we think, so the question is, why is it so common if it's so detrimental to fitness? And we think it's because it's related to susceptibility to typhoid fever. So this is a slightly a wacky graph, but it's from your textbook, so I want to show it. So they tested, this is a study that was done in mice. Basically, this cystic fibrosis, transmembrane receptor, influences the ability of typhoid fever to infiltrate the body in the intestine. And this is, if you're wild type, how many bacteria are getting in through this transmembrane receptor. If you are heterozygous, how many are getting in, and then if you have 2 cystic fibrosis alleles. And so what you can see is having the mutant allele at this transmembrane receptor reduces the ability of typhoid fever to cross through that intestinal wall and move from the gut into the body. Well, so the individuals that are homozygous for this mutant allele do great when it comes to typhoid fever. It's just the rest of life it's a bit of a problem. But these heterozygotes, it's hard to appreciate because this is shown on a log scale. These heterozygotes, I gotta get the number here because it's really impressive. They have an 86% reduction in the ability of typhoid fever, typhoid bacteria to be able to cross into the bloodstream. It doesn't look as dramatic because it's on a log scale, but 86% reduction is huge relative to wild type. So it's thought that the reason that cystic fibrosis alleles are so common out in populations is because the presence of the typhoid fever disease helps the fitness of heterozygous individuals, and therefore that keeps the allele frequency, the heterozygous advantage keeps the allele frequency in the population, even though homozygous recessive is bad. And this is a lovely figure that reinforces this. So each of these dots is a different population or different sort of geographic region, and it looks at, of the mutant alleles at this locus, what percentage are the cystic fibrosis alleles? It doesn't mean 80% of the alleles are cystic fibrosis, it means 80% of the mutants are. And so the more common typhoid fever is in a geographic area, the more likely that mutants at this locus are this mutation that prevents typhoid from entering the body. So when you see this kind of really nice relationship, that makes you think, yes, it is the typhoid fever that is keeping this allele frequency so high. Okay, so one more idea before we go on, and that is, so we think that, and I should have not changed the slide. So this is another really powerful mechanism for maintaining genetic diversity. And you may think, how can it be such a powerful mechanism if we don't have a lot of examples? We have like cystic fibrosis, we have like sickle cell anemia. There's not 1,000 examples. The reason that it's such a powerful evolutionary force is when we get out of sort of one gene, couple allele type of traits, and we start talking about quantitative traits that are controlled by many genes, heterozygous advantage is a huge, really influences genetic structure. Here you can see a goofy picture of corn, but differential strains, the F1 hybrid, look at the huge difference. That's heterozygous advantage. Every locus where the two parents differed, the F1 is going to be heterozygous, right? So if you go out and you buy vegetable seeds, you go out and you buy, you're looking for hybrid seeds. That's still with heterozygous. So really important evolutionary force, and we'll pick up the fifth one in the beginning of